What Paul showed you on this last slide was the Armsby calorimeter at Penn State, which is the first large animal calorimeter in the US. Is that right, Paul? Yeah. So I thank you for the invitation, but I don't know really why I'm on this panel. I'm not a geneticist for sure. I'm not a microbiologist. So when I need this expertise, I look for collaborators. I'm a nutritionist. My specialty is dairy nutrition. We have a small dairy herd, 240 cows, but we have the genetics of these animals, all of them. We have a good geneticist on the faculty, Chad Dekau, and he helped us with this. So I don't have any slides, so I don't have to deal with moving them up and down. But I'll just tell you briefly what we have been doing in this area. And basically, when we do these methane measurements, we are trying to look at everything, including animal genetics as much as we can with this small genetic pool that we have at our dairy farm. So we were also interested to see whether these high or low ranking animals phenotype for methane phenotype have different responses to nutritional interventions, inhibitors. We have done a lot of work with this over the years. We have probably over 15 or 1,400 cows that we have all the data, the microbiome, the genetic information and methane measurements, digestibility. We are looking at this actually right now because one of the drawbacks is that when you select for something, you select against something else. And we have to all be aware of this. Hopefully with the money that they're available now in the US, we'll have more information on this in the coming years. But we are obviously concerned with decreased digestibility when you select for low methane emissions, passage rate, and so on that. So we are looking at this data right now and looking in a meta-analysis way, and that will be published soon, hopefully. When we select, again, the drawback of having a small herd is when we pre-select, we pre-select and we measure again and we again select. And with all these different stages of selecting animals for low and high phenotype, we still end up, when we actually do the animal studies, we end up with very small differences in methane emissions, although the initial data are different. When we looked at how animals of different methane phenotype react to inhibitors, we actually didn't see any interaction, which was kind of surprising, but I think it's because of the small pool of animals that we had in these studies. But that's an interesting, I think, area to look into. As somebody, I think, mentioned that low-emitting animals may be reacting to a larger extent to methane inhibitors or other devices that we use to mitigate methane emissions. Over the years, again, we've been interested what happens with rumen fermentation, and again, if there is any relationship with methane phenotype. We have consistently seen, for example, increased butyrate production with inhibited rumen fermentation, methane production. So we did some isotope studies. There are two pathways of butyrate synthesis. So we labeled, we used acetate or glucose, carbon-labeled substrates to look at which pathway is affected to what extent, again, between high and low phenotype, methane phenotype animals. There is an abstract this year at ADSA on this. There is some interesting data there. Basically, butyrate synthesis is affected by the phenotype of the animal to some extent. Again, the pool of animals is small, so you can't really, until this is repeated and repeated and repeated again, you can't claim too much differences there. That's about the extent of our genomic approach and data. I'm sure I'm missing something, but we'll be happy to participate in the panel and answer any questions. Thank you.